Hello students, in this video I'm going to talk about chapter 7, that is excitation of skeletal muscle, neuromuscular transmission and excitation contraction coupling. So the first heading, that is transmission of impulses from nerve ending to skeletal muscle fiber, the neuromuscular junction. So highlighted from the fourth line, each nerve fiber after entering the muscle belly normally branches and stimulate from 3 to 700 skeletal muscle fibers. Each nerve ending make a junction called neuromuscular junction with the muscle fiber near its midpoint. The action potential initiated in the muscle fiber by the nerve signal travel in both directions towards the muscle fiber ends. With the exception of about 2% of the muscle fiber, there is only one such junction per muscle fiber. Now let's come to the next heading, anatomy of the neuromuscular junction, the motor end plates. So no need to read this. We have talked all of the things in the slides. Do not highlight anything in this heading. Now let's go to the next heading, that is secretion of acetylcholine by the nerve terminals. Under this heading, see the second paragraph that starts from on the inside surface of the neural membrane are the linear dense bar. To each side of each dense bar are protein particles that penetrate the neural membrane. These are voltage gated calcium channels. When an action potential spread over the terminal, these channels open and allow calcium ion to diffuse from the synaptic space to the interior of the nerve terminal. The calcium ion in turn are believed to activate calcium calmodulin dependent protein kinase, which in turn phosphorylates synapsin protein that anchor the acetylcholine vesicles to the cytoskeleton of the presynaptic terminal. This process frees the acetylcholine vesicles from the cytoskeleton and allow them to move the active zone of the presynaptic neural membrane adjacent to the dense part. The vesicles then dock at the release site, fuse with the neural membrane, and empty the acetylcholine into the synaptic space by the process of exocytosis. Now leave all the text and now let's come to the next heading, that is the structure of the release acetylcholine by acetylcholine esterase. In this, it's written on the fourth line. Most of the acetylcholine is destroyed by the enzyme acetylcholine esterase, which is attached mainly to the spongy layer of fine connective tissue that fills the synaptic space between the presynaptic nerve terminal and the postsynaptic muscle membrane. And a small amount of acetylcholine diffuses out of the synaptic space and is then no longer available to act on the muscle fiber membrane. Highlight these lines. Now let's come to the other heading, that is end plate potential and excitation of the skeletal muscle fiber. The sudden insertion of sodium ions into the muscle fiber when the acetylcholine gated channel open, they cause electrical potential inside the fiber at the local area of the end plate to increase in the positive direction as much as 50 to 75 millivolts, creating a local potential called the end plate potential. Highlight till here. Now come to page 92 and see this paragraph and highlight from the curare, that is the drug. Highlight this, a drug that blocks the gating action of the acetylcholine on the acetylcholine channels by competing for the acetylcholine receptor sites. Alright, and then there is another toxin mentioned, that is botulinum toxin. There is a bacterial poison that decreases the quantity of acetylcholine released by the nerve terminals. We talk about all the other drugs that are discussed below, so no need to highlight any other point. Now let's come to the next page, that is page number 93, and talk about the heading myasthenia gravis causes muscle weakness. Highlight this and highlight the lines which I am speaking. Myasthenia gravis, which occur in about 1 in every 20,000 person, 1 in every 20,000 person cause muscle weakness because of the inability of the neuromuscular junctions to transmit enough signals from the nerve fiber to the muscle fibers. Pathologically, antibodies that attack the acetylcholine receptors have been demonstrated in the blood of most patients with myasthenia gravis. Therefore, myasthenia gravis is believed to be an autoimmune disease in which the patient have developed antibodies that block or destroy their own acetylcholine receptors at the postsynaptic neuromuscular junction. Regardless of the cause, the end-made potential that occur in the muscle fiber are mostly too weak to initiate the opening of the voltage-gated sodium channel, and thus muscle fiber depolarization does not occur. If the disease is intense enough, the patient may die of respiratory failure as a result of severe weakness of the respiratory muscle. The disease can usually be ameliorated for several hours by administering histamine or some other anticholinesterase drug. Just highlight it till here. Come to the next heading and highlight this. The action potential spread to the interior of the muscle fiber by way of transverse tubule. The skeletal muscle fiber is so large that action potential spreading along its surface cause almost no current to flow deep within the fiber. Maximum muscle contraction, however, require the current to penetrate deeply into the muscle fiber to the vicinity of a separate myofibril. This penetration is achieved by transmission of action potential among T tubules that penetrate all the way to the muscle fiber from one side of the fiber to the other. The T-tubule action potential causes release of the calcium ion inside the muscle fiber in the immediate vicinity of the myofibril, and these calcium ion then cause contraction. This overall process is called excitation, contraction, coupling. All that you need to highlight from chapter number 7. So now let's move to chapter number 6. About chapter number 6, that is the contraction of skeletal muscle. And in the heading physiological anatomy of skeletal muscle, see the heading which is given: tighten filamentous molecule keep the myosin and actin filament in place. Highlight this heading. And the next line: the side-by-side relationship between myosin and actin filament is maintained by a large number of filamentous molecule of a protein called titans. Number 77, the general mechanism of muscle contraction. So these are the points, that are 8 points. So highlight all these. We have discussed this in the lecture as well, and we have it's also mentioned in the slides. So just mark this. This is important. Number 79, and highlight this paragraph under the heading troponin and its role in muscle contraction. Attach intermittently along the sides of the tropomyosin molecule additional proteins called troponin. These protein molecules are complexes of 3 loosely bound protein subunits that play a role in controlling muscle contraction. One of the subunits, troponin I, has a strong affinity for actin, another troponin D for tropomyosin, and a third, that is troponin C, that is for calcium ion. This complex is believed to attach to tropomyosin to the actin. The strong affinity of troponin for calcium ion is believed to initiate the contraction process. Interaction of one myosin filament. Highlight this heading: inhibition of the actin filament by troponin tropomyosin complex. A pure actin filament without the presence of troponin tropomyosin complex binds instantly and strongly with the head of the myosin molecule. Then if the troponin tropomyosin complex is added to the actin filament, the binding between the myosin and actin does not take place. Therefore, it is believed that the active site on the normal actin filament of the relaxed muscle are inhibited or physically covered by troponin tropomyosin complex. Consequently, this site cannot attach to the heads of myosin filament to cause contraction. Before contraction can take place, the inhibitory effect of the troponin tropomyosin complex must itself be inhibited. Highlight this paragraph. That is page 80, and highlight the point, the interaction of the the activation of the actin filament by the calcium ion. Highlight this line and highlight the whole paragraph from highlight this whole paragraph starting from in the presence of large amount of calcium ion till the last line, it is producing a new condition that lead to contraction. And the next paragraph, that is the interaction of the activity actin filament and the myosin cross bridges, the walk-along theory of contraction. Highlight the first paragraph. As soon as the actin filament is activated by the calcium ion, the head of the cross bridges from the myosin filament become attracted to the active side of the actin filament. Although the precise manner in which this interaction between the cross bridges and the actin cause contraction is partly theoretical, one hypothesis for which reconsiderable evidence exists is the walk-along or Rashid theory of contraction. And then the next paragraph, when a head attached to an active site, this attachment simultaneously causes profound changes in intermolecular forces between the head and arm of its cross bridge. The new alignment of forces cause the head to tilt towards the arm and to drag the actin filament along with it. This tilt of the head is called the power stroke. Just highlight till the power stroke. Energy source for contraction, the chemical event in the motion of myosin head. When a muscle contract, work is performed and energy is required. Large amount of ATP are cleaved to form ADP during the contraction process, and the greater the amount of work performed by the muscle, the greater the amount of ATP that is cleaved. This phenomenon is called the Fenn effect. So just highlight till here the Fenn effect. Alright. The amount of actin in myosin filament determine tension developed by the contracting muscle. Highlight this heading. Mark the figure 6.9 and read the paragraph for understanding. that is related to figure 6.9. All right, from this page. Now let's come to the other page, that is page number 82, muscle contraction. So highlight the first paragraph, that most of the energy required for muscle contraction is used to actuate the walk along mechanism by which the cross bridges pull the actin filament, but small amount are required for pumping calcium ion from the sarcoplasm into the reticulum after the contraction is over, and pumping sodium and potassium ion through muscle fiber membrane to maintain a proper ionic environment for propagation of muscle fiber action potential. Now just highlight the third paragraph. The first source of energy that is used to reconstitute the ATP is a substance phosphocreatinine, right? Just highlight this. Then second paragraph, the second important source of energy which is used to reconstitute both ATP and phosphocreatinine is glycolysis of glycogen. Highlight this. Then the third and the final source of energy, that is the last paragraph, is the oxidative metabolism. Highlight this 3 points only. Sorry, the next page, 83, characteristic of whole muscle tension. Highlight this heading. The isometric contraction do not shorten muscle, whereas isotonic contraction shorten the muscle at a constant tension. Just highlight this heading only, not any paragraph or not any line under this, just this heading. All right, 4, and mark this heading, the fast versus slow muscle fibers, and highlight these lines. The slow fibers are the type 1 red muscle, and the characteristic of slow fibers are smaller, and they're also innervated by smaller nerve fibers. So slow fibers have a more extensive blood vessel system and more capillaries to supply extra amount of oxygen. Slow fiber have greatly increased number of mitochondria to support high level of oxidative metabolism. They contain large amount of myoglobin, that is an iron containing protein similar to hemoglobin in red blood cell. Now the fast fibers are a type 2 white muscle. Fast fibers are large for great strength of contraction. An extensive sarcoplasmic reticulum is present for rapid release of calcium ion, have large amount of glycolytic enzymes. Fast fibers have less extensive blood supply than do slow fibers because oxidative metabolism is of secondary importance. Fast fibers have fewer mitochondria than do slow fibers. All right, just highlight the text which I have spoken. A muscle contraction of different force summation, that is under the heading mechanics. So summation means adding together of individual twitch contraction to increase the intensity of overall muscle contraction. Summation occur in two ways: by increasing the number of motor units contracting simultaneously, which is called multiple fibers, and by increasing the frequency of contraction, which is called frequency summation, and can lead to tetanization. 85, page number 86. Highlight the paragraph, the skeletal muscle tone. Even when muscle are at rest, a certain amount of tautness usually remain, which is called the muscle tone, because normal skeletal muscle fiber do not contract without an action potential to stimulate the fibers. Skeletal muscle tone results entirely from a low rate of nerve impulses coming from the spinal cord. These nerve impulses in turn are controlled partly by signals transmitted from the brain to the spinal cord, interior motor neurons, and partly by signals that originate in muscle spindles located in the muscle itself. Prolonged and strong contraction of a muscle lead to a well-known state of muscle fatigue. Studies in athletes have shown that muscle fatigue increases in almost direct proportion to the rate of depletion of muscle glycoability of the contractile and metabolic processes of the muscle fiber to continue supplying the same work output. And then read this line: the interruption of the blood flow. Highlight this line: interruption of blood flow through a contracting muscle lead to almost complete muscle fatigue within 1 or 2 minutes because of loss of nutrient supply, especially the loss of oxygen. Do not highlight anything under the heading the liver systems of the body. And then now let's come to the next heading, that is remodeling of the muscle to match function. Highlight this heading: the muscle hypertrophy and muscle atrophy. The increase of the total mass of muscle is called muscle hypertrophy. When the total mass decreases, the process is called atrophy. Virtually all muscle hypertrophy results from increase in number of actin and myosin filaments in each muscle fiber, causing enlargement of the individual muscle fiber. This condition is called simply fiber hypertrophy. Hypertrophy occurs to a much greater extent when the muscle is loaded during the contractile process. The manner in which forceful contraction leads to hypertrophy is not known. It is known, however, that the rate of synthesis of muscle contractile protein is far greater than hypertrophy is developing. Now let's come to the next paragraph. Along with the increasing size of myofibrils, the enzyme system require energy also increase. This increase is especially true of enzyme for glycolysis that allow rapid supply of energy during short-term forceful muscle contraction. All right, when a muscle remains unused for many weeks, the rate of degradation of the contractile protein is more rapid than the rate of replacement. Therefore, muscle atrophy occurs. The pathway that appears to account for more of the protein degradation in a muscle undergoing atrophy is ATP-dependent ubiquitin proteasome pathway. Proteasomes are large protein complexes that degrade damaged or unneeded protein by proteolysis, a chemical reaction that breaks peptide bonds. Ubiquitin is a regulatory protein that labels which cell will be targeted for proteasomal degradation. So highlight till here. Fibers under rare condition of extreme force generation, the actual number of muscle fiber has been observed to increase in addition to the fiber hypertrophy. The increase in fiber number is called fiber hyperplasia. When it does occur, the mechanism is linear splitting of previously enlarged fiber. Highlight this heading. The muscle denervation causes rapid atrophy. When a muscle loses its nerve supply, it no longer receives the contact signals that are required to maintain normal muscle size. Mark till here. Number 88. To death, all the muscles of the body, they go into state of contractures called rigor mortis. That is, the muscle contract and they become rigid even without action potential. This rigidity results from loss of all the ATP, which is required to cause separation of cross bridges from the actin filament during the relaxation process. The muscle remain in rigor until the muscle protein deteriorate about 15 to 25 hours later, which is presumably result from autolysis caused by enzyme release from lysosomes. All these events occur more rapidly at higher temperature. So this was all about chapter 6 and chapter 7. Integrate these video with the slides which were taught in your class. I hope this is going to help you. Thank you.